Structure of cell membrane. Introduction of plasma membrane. History. Fluid mosaic model. Chemical composition of plasma membrane. Function of plasma membrane. Introduction of cell membrane. Cell membrane or plasma membrane separate the interior of the cell from outside environment. Composed of lipid bilayers including cholesterol which maintain their fluidity under the various temperature. Selective permeable and help in transport of substance with the help of proteins such as integral or peripheral protein. Involved in the variety of cellular processes such as cell addition, ion conductivity, and cell signaling. Provide mechanical strength to the cell. History. First cell was discovered by Robert Hooke in 1665 using a microscope. Cell theory was proposed by T.S. Simon and M. J. Sheldon in the 1830s. All the living organisms are composed of one or more cells. Cell is the most basic unit of life. All the cell arises only from pre-existing cell. Most accepted biological model of the cell membranes was given by S. J. Singer and G. L. Nicholson in 1972. Fluid mosaic model. In 1972, S. J. Singer and G. L. Nicholson proposed fluid mosaic model. Fluid. Individual phospholipid and some protein can move sideways laterally in each other, therefore fluid. Mosaic. Range of different protein resting on the surface on through the phospholipid give it mosaic appearance. Just see in figure. Chemical composition of plasma membrane. Chemical composition contain a variety of biological molecules, mainly lipid and protein. Carbohydrates are present in a very small amounts, predominantly as a glycoprotein. Composition is not set but constituencies change for fluidity and change their environment. In plasma membranes, lipid nearly about 40 to 45%, protein 50 to 55%, and carbohydrate 1 to 5%. Now we discuss one by one. Lipid. The cell membrane consists of three classes of amphipathic lipid. These are phospholipid, phosphoric acid plus lipid, phospholipid, glycerol plus lipid, glycolipid, and sterol. Carbohydrates are present in very small amount, predominantly as a glycoprotein. Lipid mainly phospholipid is greater than 55%, glycolipid 2%, and cholesterol 40 to 45%. Phospholipid. Phospholipids are most abundant lipid in the cell membrane. Phospholipids consist of two classes based on the types of alcohol. Glycophospholipid and sphingolipid. Glycophospholipid and sphingolipid. Glycophospholipid consist of phosphatidylcholine, phosphatidylserine, phosphatidylethylamine, phosphatidylesterol, and sphingolipid contain sphingomycin. Plasma membranes is asymmetric in nature, consists of different types of phospholipid in outer and inner leaflet, result increase in fluidity. Outer leaflet phosphatidylcholine and phosphatidylethylamine. Inner leaflet phosphatidylserine, phosphatidylinositol, sphingomycin. Phospholipids are empipathic molecule consist of polar head and unsaturated fatty acid is tail, which is hydrophobic. The unsaturated in the fatty acid chain provide the close packing of plasma membranes. Glycolipid. Glycolipid only account for 2% of the total lipids. The fatty acid in the glycolipid usually contain even numbers of carbon, typically in between 16 to 20. Cholesterol. Cholesterol is normally found in dispersed between the hydrophobic tails of the membrane of phospholipid. Cholesterol regulates the fluidity of plasma membranes. At higher temperature, cholesterol inhibits the movement of phospholipid fatty acid and reduces the membrane fluidity. At cold temperature, cholesterol interferes with fatty acid chain interaction and act as an antifreeze. Cholesterol maintain the fluidity of the membrane. Fluidity of lipid bilayer. At low temperatures, it tightly packed, membranes are thick. At higher temperature, polar head is loosely packed, tail is disorders, membrane is thinner. Carbohydrate. Plasma membranes also contain carbohydrates, predominantly glycoprotein. Carbohydrates are located on the surface of cell where they are recognize the host cell and share information. Virus and other parasites bind to this receptor, cause infection. Protein. Plasma membranes has large content of protein, typically around 50% of membrane's volume. Types of protein: integral protein or transmembrane protein, peripheral protein. Integral protein or transmembrane protein span of the membranes and have hydrophilic cystole domain which interact with the internal molecules, hydrophobic membrane spanning domain that anchor it with the cell membranes, and hydrophilic extracellular domain that interact with the external molecules. Hydrophobic domain consists of one or multi or combined with alpha helix or beta sheet protein. Examples of integral proteins like ion channel protein pump, G protein coupled receptor, peripheral protein attached to the integral membrane protein or associated with peripheral region of lipid bilayers. This protein tends to have only temporary interaction with the biological membranes. Once react molecule is dissociated to carry its own work in a cytoplasm, some enzyme and some hormone. Integral protein. Integral protein are permanently attached to the membrane, embedded in the whole membrane, serve as the carrier protein, channel or enzyme. Detergent should be used to remove the integral protein. Glycophorin are the example of integral protein. Peripheral protein. Peripheral protein are temporarily attached to the cell membranes, located on the inner or outer surface of phospholipid bilayers, serve as the receptor and surface antigen. Peripheral protein removed by salt pH change. Erythrocyte spectrin are the example of peripheral protein. Ion channel. It is formed by integral protein. Allow inorganic ions such as sodium, potassium, calcium, chlorine to diffuse down there the electrochemical gradient across the lipid bilayers. Ion channel play an important role in controlling the electrical behavior of the cells. Nerve cell. G protein coupled receptor is a single polypeptide chain that crosses the lipid bilayer 7 times, responding to the signal molecules, hormones and neurotransmitter. G protein coupled receptor are used in process such cell to cell signaling, the regulate of product of CMP and regulation of ion channel. Asymmetric lipid bilayer. Outer leaflet, inner leaflet. Outer leaflet, lot of carbohydrate. Inner leaflet, carbohydrate does not have significant role. Sphingomycin and phosphatidylcholine is present in outer leaflet. Phosphatidylserine and phosphatidylethylamine are present in inner leaflets. Prophase is an outer direct ATP dependent transporter. Phase in an inner directed ATP dependent lipid class of transporter. Function of plasma membranes. Protective function. Form of outer boundary of the cell. Digestive. Take in the food and extract the waste product. Selective permeability. Help in transport across the membrane. Content. Cell surface receptor, glycoprotein receptors present in on RBC. Cell adhesion molecules present on the plasma membranes of the certain cell play an important role in processing of inflammation. Johnson help in formation of various types of junction, adhering, anchoring along with help of cytoskeleton element.